Hello, this is the paper presentation for Physics 177 class. Today I'm going to talk about the paper Direct Observation of Steps and Rotation of the Bacterial Flagellar Motor. What is flagellar motor? The flagellar motor is a machine that rotates the helicofilament or the flagellum that allows the bacteria to swim through its environment. Let me tell you more about its structure. The motor consists of statorines and rotorines. The statorines are a stationary part that is attached to the membrane. The rotorines are the rotary part that rotates the helicofilament. It is the statorine that drives the flagellar motor. The driving force here is the ion motive force. It can be sodium motive or proton motive. Although the process is not fully resolved, it is assumed that the ions bind to the stator and detach. In this process, statorines undergo conformational changes which allows the rotor to tick around. In other words, rotor does not move in continuous motion but rather move in units of steps. In this paper, the paper aims to resolve the step of rotation. Here, in this experiment, we use a sodium motive E. coli camera. To resolve the step of revolution, we need to track the flagellum. There are two imaging methods. One is the backfocal plane interferometry. We use a 500 nanometer polystyrene bead attached to the flagellum as a marker. As a result, we see an oval-shaped trajectory. The x and y axis is in scales of nanometers. Although it seems like the trajectory is oval-shaped, the trajectory itself is purely circular, and what we observe is the leaned projection of the circular orbit. The second method is the fluorescence method. We use a 200 nanometer fluorescence bead that is attached to the same part. We use a mercury lamp to excite the fluorescence bead, and as a result, the bead releases the photon which we can detect. However, this is not enough. Previously, the steps were not resolved due to the high-speed rotation and multiple stator units involved. Therefore, slowing down the process until only one stator unit generates the torque is needed. The way the scientists did this is by lowering the sodium motive force. They've done this by lowering sodium concentration for backfocal plane experiment or by photodamaging, thereby decreasing the membrane voltage for the fluorescence experiment. Figure 1b shows the data from the backfocal plane experiment. You see between the two black arrows that sodium concentration is decreased from 5 millimolar to 0.1 millimolar. As a result, the speed of rotation is decreased to 1 Hz, and only single stator unit that generates the torque was involved. Figure 1c shows the data from the fluorescence experiment. By keep photodamaging, we reduce the speed of the flagellar motor until it reaches 1 Hz. An interesting part here is the red arrows. We see by recovery of the sodium concentration or just by random fluctuations, sometimes the speed is doubled. This is the point where the stator unit converts from one stator unit activated to two stator units activated. Now let me show you more figures. Figure 2a shows the selected image from the fluorescence experiment. It shows a nice circular path. Figure 2b shows the angle versus time graph of both experiments. The blue traces here are from the backfocal plane experiment, and the black traces are from the fluorescence experiment. The traces with different gradient represent different average speed of rotation. The steeper the gradient, the higher the speed of rotation. Now, figure 3a is the important part. The authors applied a step-binding algorithm to figure 2b. As a result, we see a dominant step size of 14 degrees per step. Now, figure 3b shows a power spectra of the fluorescence experiment. We see that the peak of the power is where the 26 periodicities per revolution occurs. The 26 step per revolution corresponds to about 14 degrees per step. As a result, the two experiments agree in the step size. Figure 4 shows the combined data of both experiments. For figure 4a, the multiple Gaussian fit is plotted. For larger step size than 14 degrees per step is considered as a multiple of single step units. By fitting the multiple Gaussian fit, we see that the 4-step size is 14 degrees per step. We also see a backward step that is in the size of 10.9 degrees. Now, figure 4b shows the power spectra of the combined experiment data. We see that there is a peak at the 26th step per revolution, which corresponds to the step size we predicted in former data. The interesting part here is that the backward motion can also happen. This does not mean that the motor switches from counterclockwise rotation to clockwise rotation, or running mode to tumbling mode. Because the key Y protein is deleted for the experiment, the motor does not go under switching. Rather, this is because of the microscopic reversibility. In other words, this is due to the probabilistic nature of microscopic world. The sodium motive force allows the fourth step to be more probable, but still, the backward motion can still happen. Now, let's talk about the physical meaning of the result. We observed 26 steps for revolution. Previously, in the presentation, we said the rotor proteins undergo tacking motion. Therefore, it must be the geometry of the rotor proteins that are relevant to the periodicity of rotation. Surprisingly, one of the rotor proteins, called Phys, has 26 periodicity in its geometry, which is consistent to the observed periodicity of rotation. Another surprising part is that we can observe 10 or 11-fold periodicity, and they are also relevant to the periodicity of filament protein or hook proteins. Now let me show you the conclusion. The authors could have resolved the flagellar revolution. The authors resolved the direct step of flagellar revolution by using in, by making the low sodium motif force environment. As a result, 26 steps per revolution was observed for sodium motif E. coli chimera. Surprisingly, periodicity of step is directly linked to the periodicity of rotor proteins.